Thank you very much. Good afternoon, everybody. First, let me thank Libor for inviting me. It's an honor to be here. And today we will be talking about trypanosomes. And we will be talking about their beauty, but we will also meet with their nastiness because they are causative agents of lethal sleeping sickness, and they are also of a veterinary importance as they cause animal diseases affecting cattle in Africa. So trypanosomes emerged, they are ancient, they emerged around 380 million years ago, and the later evolution of humans can tell us that while wild animals are quite resistant to these parasites, humans and domestic animals are quite sensitive. Trypanosoma brucei is actually a kind of a group of 5 different subspecies. Trypanosoma brucei is a causative agent of animal African trypanosomiasis called Nagana. Nagana is actually coming from the Zulu language saying useless. And all the trypanosomes are transmitted by the tsetse fly. The tsetse fly defines the geographical location of all these trypanosomes, as they are found in sub-Saharan Africa, spreading across a vast area of 8 million kilometers squares. The human parasites, Trypanosoma gambiense and rhodesiense, they differ in their geographical locations and also in how many people they actually affect. Trypanosoma gambiense, there is 97% of human sleeping sickness are caused by Trypanosoma brucei gambiense, and it's found at the west part and the central part of Africa, while Trypanosoma brucei rhodesiense is found at the eastern and south part of Africa. There are 2 other species, Trypanosoma brucei evansi and equiperdum, and they are very unique because these parasites lost their mitochondrial genome, and therefore they became independent of tsetse fly transmission, cyclical transmission. They are now being transmitted mechanically by blood-feeding insects, even bats, and therefore they were able to leave Africa. They left the tsetse belt, and now they are found across the world, affecting mainly camels, horses, etc. In addition to Trypanosoma brucei, there's 2 other species, Trypanosoma congolense and vivas, and they also cause the animal diseases. So now, when we know the key players of my talk, I can give you a brief outline. First, we will talk a little bit about the history of sleeping sickness, about its treatment, about the latest developments and the challenges that we are facing now at the beginning of the 21st century. And I would like to conclude my talk by taking Trypanosoma brucei to the kingdom of model organisms. So first, sleeping sickness was very impactful on the history of humans. First, it has a huge impact on when slave trade emerged. As you can see here, it was recognized that slaves, black people who had these enlarged lymph nodes, they were excluded from the ships because it was obvious they would probably not survive the journey. During the colonization of Africa, the invaders were facing logistic challenges because horses were especially sensitive to trypanosomes. The first documented epidemics of sleeping sickness is dated between 1895 to 1906, and it says that around 800,000 people died. And the colonial powers were actually terrified by the speed when their working force were dying. So no wonder that they invited the best out of the best scientists to Africa to find the causative agents of this horrific disease and also hopefully to find a cure. So then here we meet David Bruce, Scottish pathologist and microbiologist. He was famous already for discovering causative agents of Malta fever, a bacteria that is called now Brucella, according to him. He was always accompanied by his wife. They would never be apart from each other for a single day, and he always acknowledged her contribution to his achievements. So David and Mary Bruce arrived to Zululand to investigate an outbreak of Nagana. So they arrived to Southeast Africa. And it took him only a few months to decipher that Nargana is being caused by trypanosomes and they are being transmitted by tsetse fly. At that time, he thought that the transmission is only mechanical. When he was examining the blood smears from infected cattle, what he noticed there are two different forms of the parasites. One was slender, handsome, another was kind of chunky, bigger, and fat. And that, as probably the stereotype of those types were saying, he thought that the beautiful slender form is the male, and then the kind of the chunky form is a female. Now we know that it's not true. Trypanosomes are unisex. And what these forms are, this is the proliferative form that causes actually the disease, and this is the transmissible form, which is pre-adapted to be uptaken by the tsetse fly. After David Bruce discovered that Nargana is being caused by trypanosomes, it took only several years to identify that also the human sleeping sickness are being caused by trypanosomes. The first description is by Robert Michael Ford from the steamboat captain the Gambia, but at that time he thought that it was a worm, the trypanosomes. But only a year after, Joseph Everett Dutton with Aldo Castellani, they said that it's trypanosome, and those are the causative agents of African sleeping sickness. And then just a couple of years after, David Bruce mentioned or discovered that they are being transmitted by the tsetse fly, and then that the transmission is cyclical, which means that the parasite is undergoing quite complex development within the tsetse fly, and it takes around 3 to 5 weeks to complete the life cycle in the fly. And here, in the meantime, also Trypanosoma vivax and condolense was discovered as well. We have two species of human sleeping sickness, but we have two very distinct diseases. So Trypanosoma gambiense and rhodesiense, they don't differ only in their geographical location, but they also differ quite a bit what kind of the disease they cause. Trypanosoma gambiense is causing chronic disease, infection that can take years, while Trypanosoma rhodesiense is causing acute infections, and the patients usually die within a few weeks. The disease is specified by two different stages. The first stage is defined by the presence of the parasites in the blood and lymphatic system or other organs. And the symptoms for the first stage can be quite mild: fever, muscle joint, headaches, itching, sore, but nothing too crazy, let's say. So this is also kind of an obstacle in diagnostics, because the patients usually don't seek medical care. But when the parasite crosses the blood-brain barrier, that's when the second stage happens. And this is already extremely dangerous. It causes neurological symptoms, confusion, disturbed sleep pattern. That's when the name is coming from. And it always, if untreated, is lethal. The first documented epidemics and also the colonial power and everything coincided, and the discovery of trypanosomes, that coincided with the onset of a modern pharmacology. So of course we have to meet the giant of that time, Professor Paul Ehrlich, the Nobel Prize laureate, who defined the theory of so-called magic bullet, which means that he says that the pathogens, the parasites, can be specifically targeted by a specific compound that will be toxic only for the parasites or infectious agents. So he was the one who discovered produced the first synthetic drug, alvasaran, that was used to treat syphilis. And also his colleagues produced the first drug, atoxyl, that was used to cure sleeping sickness. Atoxyl is actually named because they thought that it's not toxic, atoxic, but it was really toxic, and it caused blindness in a lot of numbers of patients. The new drugs that came were nothing better. Suramine, extremely dangerous and toxic. Pentamidine as well. Melarsoprol, discovered in 1949, distributed intravenously, highly toxic. The patients usually described the treatment as the fire in their veins. But anyway, there was at least some treatment for this lethal disease. So when the first epidemic was done, there was a second epidemic that started in 1920 and ended in 1960. And now with known causative agent with some drugs available, there were approaches undertaken. What is interesting that different approaches were taken by different colonial powers. The Francophones, which were dealing mainly with Trypanosoma gambiense, which is transmitted from human to human, they focused on a human reservoir elimination. So they set up mobile teams going to the rural villages, and they were looking for the patients. They were diagnostics, and they were also using the medicine, the atoxylization. In 1950s, they distributed pentamidine to 2 million people in the affected areas. On the other hand, the Anglophone colonial powers, run by English, they were dealing with rhodesiense. Rhodesiense is zoonotic. So the wild animals, the game animals, are the reservoir for the disease. So their approach was very different. They were doing vector control, spraying, getting rid of the tsetse fly. They were doing bush clearing, game destruction, and also chemopreventive of veterinary drugs. I would like to just strengthen that both of these approaches are extremely important in order to eliminate and decrease the prevalence of the disease. And this is exactly what happened at the end of the 1960s. They were able to handle the epidemics, and the number of the patients went down to, let's say, 4,000 a year. So that was a huge success. However, then something happened in the 1960s. The colonial powers withdrew from Africa, and the young African states, they were pretty much left alone to deal with this disease. The problem was that at that time, there was almost no sleeping sickness. So even though they established some national institutes to deal with this disease, there was not enough funds, there was decreased awareness of this disease. There were different health priorities. Of course, political instability, social insecurity, all of these play a role, and sleeping sickness slowly but surely was emerging. Another problem was that even though the Western world know that this is coming back, the big pharma had no interest to developing any drugs for the disease. They affect like thousands of people in Africa, rural, very poor countries. And it went up all to the 90s when we were at the same level as in the 1920s. And that time, finally, the Western world says that this is enough and we have to do something. So it was the Bill Melinda Gates Foundation, WHO, a lot of non-profit organizations. I would like to just mention here the NDI, which played an extremely important role in the eliminating of the third epidemics. So first, the DNDi came with the new combination therapy. Eflornithine was only the new drug in the second half of the 20th century, but again, it was distributed intravenously, four infusions per day for 14 days. So one treatment was 10 kilograms for one patient. And most of the patients are in rural regions when you have to take a boat. So this was almost impossible to get the cure to the patients. So DNDi developed a new, better strategy, less infusion, shorter time. But what they also did, and this is actually a very tremendous success, is that they did some data mining and discovered, let's say, 800 different compounds which potentially can be used to treat sleeping sickness. And one of them, fexinidazole, was pretty amazing. It already passed all the clinical trials. It's an oral drug, so this is amazing compared to all the intramuscular and intravenous treatment. And you can take one drug a day for 10 days, and it also is it can also cure the second stage of the disease. So here we are with the new drug potentially eliminating the sleeping sickness. So we are at this, let's say, second turning point, the change for better. But the problem is that at the end of this kind of a road, there's always some kind of a donor fatigue. There's less money. And in order to maintain the momentum, all of these have to be maintained. Diagnostics, surveillance, treatment, and vector control. We can't just now leave it that it's solved. We have to continue our effort to really eliminate this disease, and which is now affecting only hundreds of people. There is another hope, also by the NDI, acrosiborols, which are now considered as a dream drug because it seems that only one tablet will be enough to treat and to cure sleeping sickness. So we will keep the momentum, and WHO is very optimistic. They are predicting elimination of transmission of gambiense, so there will be no human-to-human transmission via tsetse fly, and elimination as a public health problem of rodenziense by 2030. So I think that we can celebrate this kind of a tremendous achievement to eliminate sleeping sickness, lethal disease. There's no vaccine against this disease, but something else happened during the last 40 years. Trypanosomes have been studied by numerous labs in order to discover pathways which can be targeted. And during those times, significant fundamental biological processes were discovered using trypanosomes, some of them unique, some of them kind of just broaden our horizon, how eukaryotic cells work. I think that we can humbly say that trypanosomes are considered now as a model organisms because they are amenable to all the genetics approaches that we know now. They are amenable to RNAi, CRISPR-Cas. We can do genome-wide RNAis, genome-wide tagging. Actually, trypanosomes are the only fourth organism that all its genes were tagged, and we know the proper localization of, let's say, more than 9,000 proteins. Do we need another model organism, you might ask? And I would say yes, because when you open a textbook of molecular biology, you will realize that all the knowledge is coming from mice, Drosophila, C. elegans, but they all belong to a very small group of Opisthocondyle. But the diversity of the eukaryote tree is much, much bigger. And unless we know how different cells deal with the different problems, we cannot really understand truly the beauty of life on Earth. So trypanosomes belong to this coba, and there is another advantage of this group. They are early branching, highly diverged, so we can even suggest evolution of certain biological processes. And this is the second part of my talk where I would like to give you a little bit of idea what we do in our labs and how we use trypanosomes as model organisms. So we work on mitochondrion, and it will be here on this picture in the light blue color. And mitochondrion is the organelle that possesses a lot of different roles for the cells, but it's most famous for being the powerhouse of the cells. It produces energy that is being used by the cell. And the energy is produced by so-called oxidative phosphorylation. The powerhouse is a very good analogy because the process of oxidative phosphorylation is very reminiscent or resemble the process of generating electricity by oxidizing coal. So the oxidative phosphorylation happens at the inner membrane of mitochondria when we have electron transport chain. So similarly, as coal is supplying the power plant and is generating as steam, the electrons, which are coming from oxidized molecules like glucose, pyruvate, amino acids, are being passed all the way to oxygen to generate water. And during that process, electrons are being pumped into the intermembrane space, creating a proton motive force. So this is the steam. When the steam is going bad, it rotates, it actually initiates a turbine, and turbine that generates the electricity. Very similarly, when the protons are being passed back to the mitochondrial matrix, it generates a rotation of ATP synthase. ATP synthase is a truly splendid molecular nanomachines that is able to convert mechanical energy of rotation to the chemical bond of ATP, the energy currency of cell. It took decades to really understand how the movement of protons are inducing the rotation and that induced the ATP generation. It took decades, it took several Nobel Prizes, but I will not be talking about this mechanism. But what else was very interesting, that during those times, the scientists noticed that the ATP synthase is being purified as dimer. That was interesting. ATP synthase are found in bacteria, it's always a monomer. They are found in chloroplast as monomers. So why in mitochondria they actually form dimer? And later on, it was also noticed that the dimers have a specific location within the mitochondria. They are found in the long rows along the ridge of the lamellar cristae. And they were also found that even in vitro, they're able to bend mitochondrial membranes. So what are cristae? Cristae are invagination of mitochondria in the membrane, and they not only enlarge the space for oxidative phosphorylation, but they also generate these microenvironments where the proton motive force can concentrate and then generate a lot of ATP by oxidative phosphorylation. So now we know that the ATP synthase form dimers, and they are always located at the rim of cristae. The advances of cryo-EM, cryo-electron microscopy, help us to understand how different ATP syntheses look in different organisms. This is all just, let's say, 5 years back. And consider the mechanism of ATP production is very similar. The structures are quite different. They form different angles. They are offset sometimes. So what is here happening? And also what we notice that the different type of dimers, which are now being categorized in 4 different categories, are linked to different cristae morphologies. And now this is becoming very important and very interesting because we can ask the following questions. Why cristae adopt these different shapes, and what is the physiological advantage of different cristae morphologies? So we will come back to our favorite model, trypanosomes, because we solved the high-resolution structure of trypanosoma ATP synthase dimer, and what we did a lot, but let's say focus here on a dimer interface. So we identified this dimer interface, which is composed of subunit G and E. And what we noticed that the same subunit G and E are also found in yeast complex, the opisthocrontal complex, but they are located laterally opposed of each other. So they do not form the dimeric interface, but they are located on the opposite ends of the dimer. But when we look how it looks in the mammalian mitochondria, the same module, E and G, is actually creating the protein-protein interactions between the 2 parallel dimers. So this is a typical opisthocrontal complex, and this would be the interaction that we observed in Trypanosoma brucei. And it would suggest that when the ATP synthase is arranged perpendicularly to the axis of the cristae, this is typical for the lamellar cristae, typical for the opisthocrontal. But when the dimerin interface is generated by the subunit G and E, and the dimers are a little bit offset, this is typical for short helical rows that we can see in discoidal cristae. And this allows us to kind of propose a possible evolution mechanism how these different cristae types emerged. So at the beginning, there was just 2 double rows of monomers, and they were interacting with each other using EG or the FO, and it depends where the protein-protein interaction was fortified and forced that lead to the type 1 or type 4 evolution, and that led to different cristae shapes and sizes. So this is the last slide before the acknowledgements. I would like to maybe just say that 2 take-home messages. First, the scientific community can celebrate that sleeping sickness is almost being eliminated, but we cannot just leave it, and we have to maintain our efforts. And second, I would like to, if you remember, that trypanosomes can be considered as a true model organisms, and using them, we can answer very interesting fundamental biological questions. And I would like to thank my team for all the work that they did. This ATP synthase project was done in foreign collaborations. I would like to thank our generous funders. I would like to thank you for your attention. And of course, I will be very happy to answer any questions you may have. Thank you very much.
Thank you. Thank you very much for this nice introduction and then smoothly moving to your own research. It's great, and I think it was in the realm of presentations at the Learned Society, so thank you very much. Please.
Yeah, thank you very much for this very nice talk. So I wonder if the interaction of the trypanosoma with the mammalian cells or the cells of the humans do a shift in the dimer connections in the human cells, and that's why the ATP production goes down, potentially, and that's why the sleeping sickness occurs in the brain? Or what do you think is the hypothesis of the sleeping sickness? It should be something with ATP, no?
So, the sleeping sickness, what is actually the cause of the sleep disturbances? No, that I probably will not know exactly. When they are, it must be, so, and now I'm not a medical doctor, so I will just try to say what I think that is happening. The bloodstream trypanosomes consumes glucose in enormous amounts. So they decrease the glucose levels in blood or in the brain. So of course they are taking the substrate for the brain to function. And they also produce pyruvate and a lot of metabolites which are being excreted, which also has a toxic effect on the brain environment. I don't know if it's directly to ATP levels. I don't know if it was probably measured, but I don't have that knowledge. But I would say that they probably affect the environment by their very high efficient glucose metabolism.
Okay. And the drugs you introduced, are they acting on the dimer?
No, pretty much there was no connections between my first thought and the second thought. I tried the transition, but no.
But actually it would be interesting because now you can find a drug working on the interaction because it's different in the trypanosomes and the humans, right?
Of course. So the original ideas of this project that I submitted 10 years ago will still be looking for the drug target because there's a lot of that I didn't say. But now it seems that we are shifting a little bit from the drug discovery to really fundamental biology. If we can maintain the surveillance and tsetse fly vector control, the sleeping sickness as such can be eliminated. But Naganas is still a huge problem, and there we can, of course, come up with some new drug targets which can be useful for treating these animal diseases.
Very interesting.
Thank you. Okay, Yula.
So now when the structure of ATP is clear, the connection with the Christie formation is strong, can you imagine, can you speculate how this could be used, for example, to change Christie, to potentiate Christie, make maybe more Christie, because we are talking about the powerhouse of the cell. So manipulating the core of the powerhouse would be extremely interesting. Can you imagine, or is this still sci-fi?
No, we are thinking along this line with our colleagues from mammalian field. If we can, for example, force the mammalian ATP synthase to make different interactions to go into the different dimer, what it would affect the Christie, the vice versa with trypanosomes. So I don't think so that's a sci-fi. I just haven't thought about the experiment that would help us to decipher. But I think that this is the way when we know more about the protein-protein interaction interface, and if we can manipulate it, we can actually, create, instead of lamellar, discoidal Christie, and then the mammalian cells will be maybe less efficient in oxidative phosphorylation, which would be my prediction, and then probably manipulate a little bit of the energy metabolism of the cells, which can be helpful for human diseases as well.
Thanks. Okay, Marek.
I have a question. So what is, just a very basic question, what is the incidence currently of the sleeping disorder in terms of the both subtypes, Rodentia and Gambiense? So it's
very small. It was a map. There's a paper from 2022 plus pathogen plus neglected tropical diseases, and based on this map, it's intense, the maximum of hundreds. So here, the red dot is just one case per, I don't know, even per year, and 11 to 30. So now the incidence is extremely low, like in units of course, it can be still underestimated because you don't survey all the people, all the patients, but also they are setting up a lot of hospitals which will have the fexinidazole available for people. So now the incidence is extremely low numbers. This is the Gambiense because it's Democratic Republic of Congo, which is the center. Rodensiense is even less. It's just we learned from the history that if we don't watch it, it will come back. And the problem is the reservoir in wild game animals. That's a problem. So we have to be still kind of watching for this disease.
So I have a naive question. So if you have them in the lab, I mean, how careful do you have to be?
Eula says that you can drink them, but we work with the Trypanosoma brucei, which is the animal parasite, and human serum lies these parasites very efficiently by complement, so we don't have to worry about that one. And when we work with Gambiense, we have to be careful, but it's much more efficient if you are bitten by the tsetse fly than just being transmitted. So yeah, when we work with the human parasites, we have to be careful, but with the brucei, we pretty much treat it as BSL-2 pathogen.
So even if one of your students is horse riding, it will not infect its own
No,
horse?
yeah, no, that's, that's a good point.
All right, okay, one more.
Are you aware of any case diagnosed here in the Czech Republic in the year, so it means the imported case?
I'm not, but I'm not saying that it didn't happen. I think there will be, if Eula knows about sleeping sickness, would have to ask maybe the tropical medicine, but I'm not aware of any human sleeping sickness infections in the Czech Republic.
So Eula, you did not infect yourself, you're saying? You infected yourself with almost all parasites? So
It's still a lethal disease, so...
Okay, so we'll keep watching. Okay, thank you very much.